Each of the dots represents one sample from the TCGA database containing more than 10,000 samples of more than 30 cancer types, with gene expression measurements and other transcription data, including more than 20,000 RNA and hundreds of microRNA expression levels. With morphing projections, the researcher can easily and interactively explore the data according to different criteria. For instance, we can arrange all the different cancer types in a circle simply by moving a slider. Let's zoom in on this group here, which contains the colon adenocarcinoma samples, or COAT for short. We can still access the individual sample data. A simple click will take the user to the sample data in the GDC data portal for additional information. We can, of course, add any other criteria, for instance, gender, and separate female and male samples. Or perhaps by race. TSNE is a machine learning dimensionality reduction technique, in which similar data points group close to each other in a two-dimensional representation. In this case, samples with similar gene expression profiles group together, creating clusters. The researcher can still separate data according to cancer type, for instance. And this information obtained by the TSNE algorithm is maintained. Let's take a look at esophageal carcinoma, or ESCA for short. We can see two distinct clusters. The application has some built-in functionalities that let the expert perform some basic analysis. For instance, we can select the samples from each of the two groups, and use logistic regression to find out the genes that are more relevant in the separation of these two clusters. Data can be exported for more in-depth analysis if necessary. On top of this, we can add more attributes. For example, separate vertically by tumor stage. The TSNE can be calculated for any set or subset of genetic information. An alternative TSNE map representation can be made using the microRNA expression, where samples cluster by similar microRNA profiles. A morphing operation between these two maps allows the user to track samples that fly away from their original cluster, or group together. Look at these small group of samples that separate from their original cluster. They group here when considering gene expression levels, but drift apart when considering microRNA. This means their genetic profile is similar to the rest of the group, but their microRNA profile is not. Let's take a closer look. The main group contains samples of pheochromocytoma and paraganglioma, or PCPG for short, while these samples are labeled as pancreatic adenocarcinoma, or PAAD. However, they are neither one nor the other. They are pancreatic neuroendocrine tumors, which were incorrectly included in the PAAD study. This means that pancreatic neuroendocrine tumors and PCPG share a similar gene expression profile, but differ in their microRNA profile. We can investigate which microRNA are more relevant in this differentiation by again using the logistic regression. By combining variables, or attributes, we can perform some interesting analyses in a quick and convenient way. For example, it is known that there is a correlation between the expression levels of microRNA 210-3P and the CA9 protein in tumors with hypoxia. We can add the expression value of this microRNA vertically, and of CA9 horizontally, and adjust the sliders. The researcher can promptly see where, in the different sets of cancer types, a correlation exists. It is possible to calculate the correlation index and p-value for any selected group of samples, so let's select this one, corresponding to sarcoma, which shows a clear positive correlation, and assign it to group A. And this one, the stomach adenocarcinoma, which doesn't, and assign it to group B. Some simple statistics can be calculated easily for these groups. Correlation between two variables is one of them. For group A, the correlation coefficient is 0.54 with a p-value less than 0.001, showing a weak, but relevant correlation, while for group B it is just 0.13, with a p-value of 0.011, showing no correlation for this cancer type.